Hello, I'm Tad Dowles, assistant professor at University of South Carolina. This talk was presented at the Ecological Society of America conference in 2022. I think it was in a section of metapopulations. I'm actually not sure now. It's been a little bit too long. But thank you for coming to the talk. This is sort of some collaborative work with me and some graduate researchers in the lab, Lauren Holian and Cleber Ten Caten. And this was funded by the National Science Foundation as part of a macro systems and neon-enabled science call. And so I'll talk a little bit about that. But my interest in metapopulations has sort of stemmed back from, I was a postdoctoral researcher working at the University of Helsinki. A lot of the colleagues that I worked with were formerly part of the metapopulation research group, sort of founded and led by Ilkka Hansky. And so today I'll be talking about abundance-occupancy relationships, which are related in a lot of ways to a lot of the sort of metapopulation theory that Ilkka was pushing forward in those terms. And so these basically posit that more widespread species should also be more locally abundant. This can be examined in two different ways. And so on the left, we have the intraspecific case, where we consider one species sampled through time, represented by that single deer icon. So you measure its abundance, you measure its occupancy. Occupancy defined as the fraction of sites that you've sampled where you find the organism. And then abundance is just the mean number of organisms that you find. And on the intraspecific case, it's not the mean, it's just abundance. It can also be viewed, these abundance occupancy relationships, interspecifically. And so there, you're looking at a bunch of different species, often at a single point in time. And so you're saying, at this point in time, we have this fox. It was present in 90% of the sites that we sampled, and it had a mean abundance of whatever, some high value here. And there's a general positive relationship in both of these cases. But when we're estimating occupancy, we're sort of inherently limited by which sites we choose to sample, right? And so here we have a map of the US with black points, if you can see them, corresponding to these sampling sites from NEON, which is the National Earth Observatory Network. Really great data, but I mean, spatially limited in a sense when we're asking questions where number of sampling sites and distribution of sampling sites can introduce sensitivity in terms of the overall relationship we observe. And so this figure is from a recent Global Ecology and Biogeography article that was led by Cleber Ten Caten, a researcher in the lab. And so this study that I'll briefly go over use these NEON data. These NEON data consisted of a bunch of different taxa, beetles, birds, fishes, you can read there, sampled across a collection of aquatic and terrestrial sites. And the goal with this study was actually just really focusing on interspecific abundance occupancy relationships to gauge support across all of these taxa and across all of the time that the NEON data were sampled for, just to gauge support for how strong the relationships tend to be. And so for each year, we measured abundance occupancy relationships, and for each taxa. And so here, each box corresponds to a taxa. The slope coefficient, basically how positive the relationship is, corresponds to each one of those points. Point color, we can sort of ignore for this case. Really, the main part about this figure is relationships tended to be very weakly positive, where that vertical gray line corresponds to the standardized slope value of 0. And so relationships tend to not really change over time, like they're not really increasing or decreasing, and they tend to be quite weak. And so this is evidence for interspecific abundance occupancy relationships, but they're weak. And they also don't explain much variation. And so here we're plotting out the R squared, the coefficient of variation. How much of the variance can we explain in abundance just by knowing something about occupancy? And so we have all of our taxa here identified by little cool silhouettes. We have our years here, and then we have our different sampling grains, which we don't really need to worry about for the sake of this talk. And we see like there are some instances where R squared gets above like 0.4. So R squared is bounded between 0 and 1. But most of the time, it's fairly low. Most of the time, it's like we're not able to explain much of the variation in abundance as a function of occupancy. And that's not really that great. Like, we call this a macroecological law, and yet this is sort of what we have. But this sort of empirical macroecology is super duper useful, but it also generates just so many questions, right? So why are the relationships so weak is like the question that immediately springs to my mind. And there's a number of reasons it could be. And so interacting species really means that you have to consider the community context, fluctuating environments, land use change, species that specialize in certain habitats, as well as just the classic specter of the detectability and sampling issues. The distribution of the sites, when you choose to sample, how you choose to sample, all really matter. But what if this stuff didn't matter? What if we could construct completely artificial landscapes, simulate dynamics, and then test to see if we can see support for these abundance occupancy relationships in sort of simulated systems where we can control all of these fun bits, or we can control the dispersal pathways between sites, where sites here are circles and dispersal pathways are these lines connecting these circles. And so in the simulation, I'm not going to go into too much detail. It's a very simple sort of population dynamic model. So each one of these circles is a population. Populations are allowed to grow at some rate R, that's the population growth rate, and they're constrained from just growing out of bounds and growing exponentially or geometrically by this alpha parameter, which sort of captures intraspecific competition. All right, so the way I'll set this up is we can start from entire neutrality. We can assume that all individuals are equal, all patches are equal in terms of population growth rate. We'll simply randomize the initial abundance and seed a certain number of these populations and the spatial networks. So we generate a bunch of different spatial networks because we actually really wanted to sort of explore how landscape structure influences these relationships. And this is the absolute baseline case. Like really the only thing that is going to affect overall abundance occupancy relationship is going to be the influence of stochasticity and then maybe potentially some dispersal limitation depending on network structure. But we can add different layers onto this. And so we started from that base assumption of absolute neutrality, but now we can actually, let's let species differ in their population growth rates. That's a good idea, right? Because species do differ in population growth rates. And so here now we randomize initial abundance across sites, and we also randomized occupancy. Oh, and so that's the actual baseline. So we actually haven't in that last slide changed population growth rates. This is when we change population growth rates. So we're just really incrementally building up this model. And so the previous slide, this change in randomizing initial abundance and occupancy starts to get a dispersal limitation and sort of that aspect. And then now we're just starting to incorporate differences among species in this iteration of a null model, I guess we can call it. And now we can start adding in some covariance, which I think is the really interesting part. What I mean by covariance is that we can allow this population growth rate to vary with things like initial occupancy. So if you have, as a species, a really high mean population growth rate, let's start you off with a higher initial occupancy. That doesn't mean that you won't go extinct, right? In some patches, it just means that species that have a higher growth rate are more likely to occur in more of these habitats, at least initially. And so you notice that we're already mashing in a lot of assumptions that are like, well, that should produce an abundance occupancy relationship, surely. And we can go one step further. And now we can, and actually I think there's one step beyond this. So this next step, this step is that we allowed dispersal probability to positively covary with not only initial occupancy, but also dispersal probability. So any of those dispersal limitations where a site could be occupied and it could support a viable population, but the individuals just can't disperse there, it's too far away. That barrier has been eliminated by working in this assumption. And no, those were the 5. We had 5 conditions. I gave this talk too long ago from when I'm recording it now, and so I'm already forgetting. This work is it's actually not in review anymore. It got rejected, but we're making some adjustments, and we'll be resubmitting shortly. I still think it's very, very cool. And so we can simulate a bunch of these dynamics across a whole lot of landscapes, and we can look at both intraspecific abundance occupancy relationships and interspecific abundance occupancy relationships. Here, the intraspecific relationships were assessed after, I think we had like a sort of burn-in, or we removed the first 100 time points of the dynamics and then used the last, I think it was 300 or so. So these are sort of longer term simulations. And we see very weak relationships. And that's sort of to be expected. It doesn't change as a function of all of these different assumptions that we've built in. Intraspecific abundance occupancy relationships were weak in our simulations, regardless of what we baked into the underlying model. However, we see interspecific relationships, where each line here is a community generated without any sort of interspecific interactions. So competition is not playing a role here. That's one thing the reviewers didn't really like, but try parameterizing the competition matrix when you have a 50 species community. I would argue that you have to make more assumptions there than I'm willing to make. And so we see this in this completely null model. We see some positive relationships that are significantly positive, but the relationships are really all over the place between occupancy and mean abundance. However, as you start building up more and more of those assumptions, we see a clearer relationship. Here, dashed lines are those non-significant ones, and any colored line represents a significantly positive relationship. And as we start to incorporate more and more species differences and more covariance between demographic rates and dispersal capability, we see more positive relationships. And this makes sense, but is also still quite interesting because it allows us to start to explore exactly which conditions and which covariances lead to these strong relationships. We can also start to explore how the size and the connections of the patches in these spatial networks start to matter. And so we found one thing that's interesting is that network size seemed to have differential effects, where intraspecific relationships tended to be sort of negatively related to network size. And meanwhile, interspecific relationships tended to be, in their strength, positively related to network size. And so it makes sense when you consider interspecific relationships and the importance of dispersal limitation and those far off patches. But you see it even when we assume complete neutrality. And so I think it's really, really interesting. And this is still sort of ongoing work in which in the manuscript, we don't dive as much into the aspects of network structure as I think we could and we should and we will. Because I thought the main thrust of the manuscript was on those the different nulls, basically, the different variations and demographic covariances that we stacked on, as well as just the intraspecific and interspecific bits. I sort of ignored the network stuff and put it in a supplement. It'll be changed before. And I'll share the link to the paper when it gets published. So you can see the final product. This is very much also still an ongoing work in progress. So basically from this short bit, what have we learned? We've learned that for interspecific relationships from Cleber's study, that in empirical data, they tend to be actually pretty weak. Or no, yeah, that was interspecific. They tend to be fairly weak, quite weak actually. But in our simulations, even with a tiny bit movement away from neutrality, as we started to get those demographic trait covariances between dispersal ability, initial occupancy and population growth rate, we see they're quite strong. They're expected to be very, very strong, and yet we don't see that in nature. However, for intraspecific relationships, we see they're quite weak, both in natural systems and also in our simulations. Not saying that they're weak for the same reasons, of course, right? If you recapitulate a pattern with a model, it doesn't actually lead to any sort of mechanistic explanation of the natural phenomenon, but it is interesting that both of these relationships, sort of in intraspecific relationships in empirical systems and in simulations, tended to be quite weak, despite this really being called like a macroecological law. And so zooming out, wrapping up, big picture ideas, these abundance occupancy relationships are weak and not predictive. We should not use occupancy as a predictor of abundance. It's a really bad idea. Yeah, just full stop. I think that there's a lot of interesting questions and open avenues for exploring macroecological questions using simulations to set up, just in the simplest case, like provide a simple baseline for what we expect to see given this long set of assumptions. It can provide a lot of cool pathways forward. And it's important to note that, and this is for the reviewers of the manuscript, models will rarely include and should not include all relevant drivers. If you build a model that has all the features of a natural system, you have a very over-parameterized, non-generalizable model. Don't do it. It shouldn't be done. Even in the best case, it becomes like an analog system, right? You get exactly like out what you expect when you mash everything in. Yeah, complexity doesn't come about by just like mashing in as many parameters and as many interactors as possible. I digress. This is already a bit longer, I think, than I should have been talking. And so with that, I will close, acknowledge my funding sources, acknowledge you for watching this. Thank you very much. And I will stop my share and stop recording. Take care.